We
This
already
conference
started
will now be
processing.
recorded.
Okay, yes, note to everybody, the session is being recorded. And what I wanted to say is just a review of the course. We have already learned how to process next generation sequencing data going from raw data to an expression table. And we already did differential gene expression and understood the statistics of t-test. What exactly are we trying to measure? Now, let's do a quick review of RNA-seq. As you remember, we talked about the significance of pre-processing, how do we remove adapters and remove PCR duplicates. And then we talked about the mapping, different strategies for mapping, including mapping on known transcripts with bow tie to T, mapping on genome with top hat and high sat. And then we talked about a combined strategy where we incorporate known transcripts and newly discovered sequences that are being expressed. And then we finalized that with quantification methods, when we looked at different ways we can quantify the level of expression and count the reads that are mapped to a specific area. This was the next pipeline that we did. So we actually, as you can see, the color coding is the same as the, just one second, Sahil, I can hear that. Okay, so what we see here in terms of colors, as you can see, the blue is pre-processing, the red is mapping, green is quantification. So let's take a look over here. This is, sorry, the differential gene expression. So you can see the blue, we started here, and this section is all blue, so that would be pre-processing. And then we have the mapping strategies right here, where we have top hat 2, and high sat, and we also have bow tie to G, BWA, and star. These are different ways to map on genome. But once you've mapped on genome, you have to do 2 additional things. You have to map the exons, because as you remember, we only look at exons, and typically in alternative splicing, the introns are being cut out. So that's why we have the exon detection. And then we have to identify junctions. And junctions are those areas where exons meet together, and we can see a single read that has the end and the beginning of an exon. And that's how we know that those were spliced together. So that is mapping on junctions. Now, top hat and high sat, they actually do those things internally in a single algorithm. After those have been done, we have to construct isoforms. What are isoforms? Isoforms are those transcripts that we've identified where we see the alternative splicing and the exons merge together, and different numbers of exons and in different orders could be combined into a transcript or an isoform. So you have this step of isoform construction, and then we process that data and we prepare a new reference file. The new reference file includes all of the mapping that was done in this step and the isoforms that have been identified. After that is done, now we have bow tie to T that will use this GTF file and align the reads to the newly identified transcripts that are placed in the GTF file. After that, you have to count them. And so the green represents the quantification step, where we count the number of reads aligned to a specific region. Now, as you remember, we talked about different ways of counting. We talked about FPKM, fragments per kilobase per million. We talked about TPM. We talked about just raw count. So there are different ways to count those. And then finally, we discussed differential gene expression, where that idea of students' t-test, of t-test statistics, is applied to a comparison between 2 groups, where we look at p-values and log-fold change that represent the difference between 2 groups. Now, let's go quickly. And for those of you that did not have a chance to do it last time, we will build that pipeline together. So what you can do is go to areas of analysis and go to RNA-seq parallel analysis of NGS and microarray data. Kritika, I know that you haven't started this, so specifically this is to help you get this done. And if any other users here have not started it, let's try and do it together. And tell me if you have any questions as we are preparing the pipeline. So the same file that we had from before could be used. So let's first set up what we remember about the file that we have. We have model genome, right? We're using human samples, so we will keep this the same. And we are using pair and reads. And we have FASTQ files. And here we have to select the latest assembly, GRCH38. Now here, we do not upload the files from here. We use URL bulk upload. And we have a sample SVL called samples SVL. And just for those of you that would like to have that file for this session, I will give you the link right now. Just to remind you what this is. Okay, so I'll give you the link. Okay, so what is inside the samples.svl? Let's just quickly take a look. These are SRR, so sequence read archives, and they have a number of samples, 11 samples to be more specific, but they are paired end, so that means the reads were read from both directions, and that gives us this _1 and _2. So we do URL bulk upload, and now we do have to define groups. So to define groups, we actually have to create 2 groups, and we have to look at which files belong to which subtype. So we have 2 subtypes here that we want to compare, non-malignant and cloud and low. So you can see that we have, let me make this a little bit bigger. We have the names of the cell lines, and then we have what subtype they belong to, subtype of breast cancer. And here we have those names of the actual archive. So let's go in here and try and drag and drop in the right folder what we have in the right group. So what I will do is actually place it right here. Okay. Okay, so the first file, one sec, sorry. Okay, so SRR 925720, so let's find it 720, 721. These are non-malignant or normal-like, so I will place them in group 1, in group A. 634 is also non-malignant, and 634, 687, and 688, as well as 697. So this one. And the rest are going to be cloud and low. So by placing normal-like in group A and cloud and low in group B, I have to remember this because later on I will be looking at those p-values and log-fold change, and I need to understand what is being compared to what. Now I press continue, and now for contrasts, I can just do select all, submit all changes, and now I can build my pipeline. So the first section that comes up, again, we can skip this step of pre-processing just to speed things up, but we will have a whole week for this pipeline to finish, so we can go ahead and include those this time. So we can select Trim-O-Matic, and then we can do PCR clean. And then we can, after we complete the pre-processing, now we can do either mapping on genome, right? So that would be one strategy. and then we have to do all the steps, or we can just map on known transcripts. I will do mapping on all transcripts. You can choose whichever pipeline you would like to do. So I will go to bow tie to T, and then RSM expression table. And now I have this option to select one of the differential expression methods. So I can select dsec2, which I did last time. So this time I will actually select edge R. So now I have my pipeline, and I can do end, save, and let's save this pipeline. So this would be cell line, and maybe just to remember, normal, like versus cloud and low, edge R. Run pipeline. Okay, so we started this pipeline in alternative to the pipeline that we did last time, and we'll see next time what would be the difference in the results. Okay, does anybody have questions about this pipeline, or should we move on? Okay, if you have any questions, type them into the chat box. So now we have the results of this, of the dsec2 from last time, and this time I started edge R, but the results essentially are going to be very similar. So as you can see, this is a screenshot from that table from the results, and I will show you those results when we download them, and this would be the result from edge R. Now both of them contain a fold change column, so log fold change here, log 2 fold change, but essentially the same thing. And then here we have the p-value, just like here we have p-value and p-adjusted value, and here we have p-value and FDR. So essentially p-adjusted value are these columns combined. So let's take a look at what this actually means and how many genes do we have in terms of our differentially expressed genes. So in the zipped file that I gave you, or in your pipeline, if you completed the pipeline, you will get something like this, a zipped folder, dsec2 all files, right? So let me just show you under my pipelines. And let's go to the previous one. This was my pipeline. What I downloaded was this dsec2 all files. So dsec2 all files contains, again, the different ways to count the differentially expressed genes, as well as isoforms. And what I can open is fpkm dsec2. So if I open that file, it will look like this. So I have log 2 fold change. This was the raw file, so I'm just going to let me remove all of the filters here. So this is what I get in the beginning, and what I want to do is I want to select those genes, remember these are genes, that have a good p-adjusted p-value. So what I would do is I would add a filter here, and first of all, I am looking for a p-adjusted value of less than, so less than or equal to 0.05. So if I sort or filter out just those genes, I'm going to select all of them. And let's just create a new sheet and place them here. And let's take a look at how many of a good p-value do I have. So I only have about 1,000 genes with a good p-value, but I'm also interested in the fold change, log 2 fold change, being significant. So what I want to do here is, again, filter and select only those that are more than, greater than, or equal to 1.5. So now let's see how many I have. So actually, let's just, again, select all of these. and place them here. And now I have 230 differentially expressed genes. To understand what differentially expressed really visually means, I can take one of these genes, right, and I can go back. So in the same results, I have the RSEM results. So I can, for example, download expression genes FPKM, and this file would contain just the original expression values, right? So I can open this file again in Excel, and we can see here that this was with the pre-processing steps. So it included here no adapters, and no PCR here at the end. So I can just remove this from the header, etc. Right? And I already have this table prepared and given to you guys. So this was the expression genes, right? And now I can find the gene that I selected. So let's go back to our differentially expressed table right here and try and find this gene in here. So we can search. Okay, the first one was not found. Let's see some other one. Because I filtered this table by those genes that were insignificant. So let's try and find it. This one is also not available. Let's try a different one. Okay, maybe there's some different other files. So let's just make sure that we've got the right thing, and I'll. Okay, so I already have this open here. So that would be right here, right? So let's just finish removing this real quick. Okay, and let's try and find the gene. So at least we know that it does exist in this table. There we go. Okay, so the reason I was cleaning up the top headers is because we actually want to see if the difference is really between the groups that we selected. So if we put here a bar plot, we do see that there are some differences, but it's yet unclear between what and what. So let's go to the top and finish editing this and just for ourselves identify which ones are normal-like and which ones are cloud and low, so that it will be easy for us. Now you can see that these are all in group 2, right? So let's see, it should have placed them in according to what we selected in the groups. So let's see, 720, that is normal-like. 688 and 687 are normal-like. 721 and 34, these are normal-like. 36 and all the others are cloud and low. So now let's find that gene again, and what we can do here is just freeze the top row just so that we can see properly which ones are which. So let's take one of the examples. So the first example that we had was this one, and we can select, insert a bar plot, right? And so we can clearly see the difference between these and these. Let's just make sure that we annotate the horizontal axis. And now we can see that 634 is where the normal-like end. So all of these are normal-like, and these are cloud and low. Now for this gene, 4139, if we go back to 4139, that was the first one, what are we seeing here? So let's just take a look over here and copy this and go back here, and let's place it in here. What does this mean in the context of the actual expression? Okay, so what we see is the log two-fold change is about 4, very good. P-adjusted value 0.003, so statistically significant. So if we take the averages, right, you remember what we did previously. So let's just do over here t-test. So this would be average of group 1 and this would be average of group 2 and this would be fold change. So let's take average of, this would be our first group, and then this would be average of the second group, and the fold change would be this divided by this. So 1.44. Remember that we were looking at, so if we now do (0.15), right? What if, so what we are looking for, right, is, okay, log 2. So let's just do, okay, yeah, log of this. So remember when we talked about these two values, we talked about doing fold change in 2 different ways, right? So let me copy just the values. So I want to just do values. And I can also do this minus this. And now we will take a look at log of this. So these are some fold changes that we get with t-test statistics. So the question is, why is it so different, right? So when we take a look at 4139, okay, so first of all, what did I get here? Oh, okay, so I'm not working with the right gene. Let me quickly, 4139, where is 4139? Okay, it's right here. So let's take a look at that for 4139, right? 4139 is this one. Okay, so let's take first the average. That would be 43 to 43 and 43 to 43. Okay, 1.4. That's much closer to what we were expecting. So we can delete this. Okay. So actually, we now see so log fold change was 3.95, right? And the p-value was very good, and the p-adjusted value is even really good. So this is actually a good representation of differentially expressed genes. Still, as you can see right here, you have actually this sample, probably according to just the differential gene expression, belongs over in this group, right? So the conclusion from this analysis is that differential gene expression could be a powerful way to compare two groups, but it has some limitations. And the limitations are that there's a variability within this group and a variability within this group that is not really represented in this analysis. So when I want to look at the data that I have and base my biological interpretation on the data, it is very important to first adjust the data so that it stays consistent and more useful for these types of analyses. So what we want to do today is take a look at how do we kind of give an overview of the whole data and understand what our data looks like before we jump to conclusions using this type of analysis that could lead us to sometimes confusing interpretation that, on average, this is true, but for individual samples, there might be a very big variability. And so I might be, accounting for various technical variations that I do not understand. So if we go back to the presentation, I want to talk to you about transformation and normalization as a step before the actual analysis. So let's take a look at this visual representation of the data. Does anybody know what this is, what this type of visualization is called? And what does it show? So does anyone know? Okay, great, box plot. So do you know what are these bars in the middle? What do they represent? Okay, so since, maybe, yeah, go ahead. Somebody wants to answer? Okay, so if, it's not very clear what this plot does, it actually talks a lot about the types of things that we were just looking at. The middle here is the median. So this would be if you take all the values and you find the median of values in a condition, like here we have two conditions, two groups, and this represents four groups. We would want to see what is the average. So kind of what we rely on for t-test, only it wouldn't be the average, it would be the median. And we also have different quartiles that helps us understand how is our data structured, right? So just to review the elements, we have the max value, and then we have the upper quartile, we have the lower quartile. So this is the bottom 25%, the top 25%, and this is the middle 50%. And this would be the median. So if you just take a look at this box plot, you right away spot outliers, which are not typical for the data represented in this condition. And you kind of see the standards within this box plot. Now, if we apply this idea to our data right here, how would we do this? What we would do is we would actually take all of these samples. Well, before we do that, let's give them a different name so that in our box plot, we can actually see that very clearly. So let's insert a row here. So let's insert a row here. And we'll call this normal like. And this one we'll call cloud and low. So now in Excel even, it's very easy for us to do a box plot. And let's take a look at what it shows. So surprisingly, what it shows is something very different from what we observed, right? It shows us that actually the majority of the values are outliers. And there are a lot of outliers, but the majority of the values are towards the lower end of the values that we observe, which means that we have some highly expressed genes, right? But the majority of them are actually very low expressed. So there is a huge difference between these very big values and the majority of the values. To, first of all, address that problem, we have two strategies that we can use. The first strategy is to use logarithm scale. So what would logarithm scale do? Let's try and do that really quickly so that we understand what logarithmic scale really looks like. So I will take my table and create a new section here, and this I will call logarithm scale. So what I would do is I would just create a formula. I would take this number, right? So actually I would do logarithmic scale of this number. So now I can replicate the names, and I can just go ahead and do that for all of these. Right, so I can now just drag this, and as I drag, it will show me all of the values. But when I have zeros, you cannot take a logarithm of 0. So instead of doing that, my formula should have something else. I should actually add 1, because what is logarithm of 1? Is 0. So if I add 1, it's actually not going to change my original value, right? So what I do is I add 1 to the original value, and then the log of this would be the same as log of this, unless this is 0, right? So now I can do the same thing. I can actually, well, let's delete all of the other ones and delete this. Okay, so now you can see for this one, it was 0, it remains 0, right? And all the values continue. So I can take this. Oh, okay, so that's why. Okay, so what is this? This should be, again, logarithm scale of this plus 1. All right, so now I have the bottom and the top, and I can copy this and go all the way down and paste. All right, and now I can just stretch it out for all of my samples. Right, so 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. So now I have logarithmic scale. How does logarithmic scale affect box plots? Let's insert a box plot here and take a look. Now we can see that the values look more reasonable, right? So we can see that the values still have variation, right? We haven't really changed it that much, but we do see that we observe more of those smaller values, right? Which means that they will have a greater significance in our downstream analysis. So logarithmic scale is very useful for us. And logarithmic scale allows us to really appreciate those low values and the high values and kind of stretch it out so that we can see a greater detail of what's going on in our data. Now, the other method that I want to talk about is called normalization, specifically quantile normalization, right? So let's go back to the presentation and I will show you some of these methods in detail, what they do. So we talked about the box plots, and then we prepared this table for you already, and we see that these numbers could be converted, right? So we talked about this in logarithmic scale being a very simple formula, the formula being logarithmic value of this plus 1, right? Because we want to avoid the zeros. And now we can run descriptive statistics of these different data sets. So before we look at the median and the skewness, let me show you how to run normalization, and then I will share with you the table that has the raw data, the logarithmic scale, and the quantile normalized data. And we will start comparing the summary statistics of the data and see how they vary. Okay, so let's go to the platform. And in the platform, under areas of analysis, you have this area called utilities. So let's go there real quick and let us upload the file that we just recently downloaded. So we downloaded this expression genes FPKM. So let's just choose that file. We do not need groups. And then we see quantile normalization right here. So let's take quantile normalization. What does quantile normalization do? It actually aligns the averages or the medians between the samples and keeps the distributions to be similar between samples. So in logarithmic scale, we saw that kind of stretch things out. And then, so right here, you see transform table to ln. So we do want that. And then we wanted to stretch things out like this. So we actually don't need a threshold here. Threshold would prevent some normalization for example, low expressed genes. We can use it. For example, you can set it at 5, or you can keep it open if you want to address all of the values that you have. Now you have the option to connect it to PCA, which I'll talk a little bit in detail next, but we won't do it right now. Let's just save it as expression quantile normalization. And we can run the pipeline. Okay, so I don't want to leave anybody confused about this. And I know that this is maybe a little bit different from what we did before. So I just want to take a short pause here and ask and make sure that everybody's following and you kind of feel comfortable with what we're doing and understand the significance of these things. So does anybody have any questions? Okay, so feel free to type it in. I'll address your question. In the meantime, you can see that it's processing. And I want to show you that this file is already going to be available for you in the in what I shared with you in the zip file. So you can actually take a look under lecture 4 files, that zip file that I gave you. And inside here, I have this Excel sheet, expression genes filter 6. So I actually already have it open. Okay, expression genes FPKM filter 6. What is in this Excel file? In this Excel file, all of these tables are already prepared for you. So you can see that we have the original raw data. So this is normal raw data. And then there's t-test and log fold change, and how many differentially expressed genes you can observe using just t-test. Then you have a logarithmic scale, right? So this is ln C2 plus 1. And then you have a smaller number of differentially expressed genes, right? And then you have normalized, quantile normalized data over here. We'll take a look once it is prepared, what it looks like. But really, the results are here. And again, you have differentially expressed genes, and you have a much smaller number of those, right? So this is what the comparison of differentially expressed genes with the same threshold would look like. So it dramatically decreases from raw to quantile normalized data. Now, let's take a look at the summary statistics. Okay, so let me just check that my pipeline is running. It's done. Let me know if yours is done as well. But if I go in here, I want to download pipeline output files, and you can see here that the normalized file is right here. So if I download it, and I look at my normalized file, okay, I can see that the numbers have changed. So you can take a look at that. Now, how do we do summary statistics? How do we get to this kind of a table, and what does it actually mean? So first of all, let's take a look at the box plots that we just looked. So this is the box plots for the original data, right? We saw the same thing. All of the values were at the bottom, and then some outliers were plotted right here. If we did a logarithmic scale, the data looked something like this. So the lower values were stretched out, but we still have this lower quartile going almost all the way to the very bottom. And you see some variation between the medians right here. What happens if it's normalized? Well, actually, if it's normalized, as we expected, the distribution was normalized, and these medians were perfectly aligned. So now this data, why is this data better than this data? Well, it's better because when we compare samples, right, when we compare the samples and the differences between them, we want to adjust the comparison for the overall distribution of the data. And the distribution of the data could be aligned in the middle, right? So this alignment in the middle doesn't mean that we actually change the values. We actually transform the data so that they align here, and the rest of the differences are either going up or down from this middle point. And so if we take a sample from here and a sample from here, they are more comparable, like apples to apples or oranges to oranges, instead of comparing completely two different things. So we are putting things in the context. And in the context of this experiment, now this data is much more appropriate to be compared, especially when we talk about values of genes being expressed. And expression is, of course, reflective of what's going on in the organism, as opposed to being a universal value, right? So this idea that the biology of this group of cells or that group of cells represents something completely universal is not so true because we actually have to put it in the context of the biology of this specific sample. So this is what quantile normalization actually does. So now let's, using this understanding, again, what are we looking at here? Well, we're looking at the median of each sample being aligned, and we are looking at how is it distributed. So is it, what is skewness, for example? Skewness is whether a distribution is off to the top or the bottom, for example. Or, you know, averages would kind of be close to where this line is. This is the median, but average would be close to here. So those are some of the things that we can also visualize and see using summary statistics. So if we take the differentially expressed genes, let's take a look at this normalized table. Or actually, let's just take the raw data that we have and run some summary statistics. So what I can do is I to data. Now, not all of you, if you're using Excel, would have the standard package for summary statistics. So it's you know, important just to add that if you don't. And I think it would be under Excel add-ins. And here you have the analysis tool pack. So if you click on data analysis now, you can see a number of things that are possible, like t-test, regression, histograms, descriptive statistics, and others. And what we want to look at today is descriptive statistics and what we can learn from the overview of data. So if I take all of my data here, and labels are in first row, of course, and then I can put all of my results into a new worksheet. Okay, and what do I want to do? I want to actually get my summary statistics, right? So we'll take a little bit of time, and now I got my summary statistics for each sample. Now what you will notice is that this is a very easy table to work with as it comes out. But if I move this, I will have a repeat column, so I can just delete all of these, and then it will be much easier for me to see because now these are aligned under sample. So what do I see here? I see the median, right? And so I can, for example, insert a bar plot of the median. And I see that the median value differs between samples. I can also take a look at skewness. And now I can see that actually they're not all distributed evenly within each sample. What happens if I do the same for my normalized data? So now as you understand what exactly this table is showing and what we did to obtain these numbers, let's go back to the prepared table and let's take a look at this tab, Summary Statistics. So how did things change after we did normalization and logarithmic scale? And you can see that logarithmic scale actually is very close to quantile normalized data. So let's take a look. These were the original mean values. You can see the variation. And these are now the adjusted mean values. The same thing for, this is skewness, and this is the skewness that is adjusted. So what will happen if we remove here those, you know, it's hard to see exactly what's going on because of the green bar. So let's just remove it. So we don't want to look at raw data anymore. Okay, and let's adjust this value as well. So let's change it to maybe 3. sorry, maybe 1. Okay, so this is what you see that quantile normalized, actually skewness is even. Logarithmic scale, what does this show us, right? So 687, 688, 720, 721, 634, what are these? These are actually the normal-like samples versus the cloud and low. So they're distributed differently. And obviously, just because of the distribution, we will find the t-test statistics, which assumes equal distribution within those groups of samples, will give us some misleading results, okay? Okay, concerning the question for real-time PCR, so real-time PCR measures things in different values than RNA-seq. So it's a way to confirm if you observe values that are, they have the significance and they show some fold change or, some difference. It's a confirmation of what you see in RNA-seq, but the values themselves are different, if that makes sense. And also how we measure genes is also different from, so RNA-seq maps it on the genome or uses a reference database of known transcripts, and RT-PCR relies on however you prepare your, libraries, right? Yeah, so again, different values and what exactly you're measuring is a little bit different, but comparable. Yes, you can compare, but it's not exactly the same. Does that make sense? Okay, so just in the meantime, Develina, let me know if that makes sense, or we can talk more about what that would mean. But as you can see, that you
Hello.
have, yes.
Hello. Yeah, actually, I have some data of RT-PCR and of particular genes, and I wanted to compare those gene fold change value with the TCGA data of HNSCC, which has got the same kind of gene set. So I was wondering if I could compare those things, like, as because that is a completely bioinformatics based data, and I have a wet lab data. I wanted to validate the TCGA data with my wet lab data. So is it possible?
Yeah, so again, the significance would be comparable, right? If you look at, the fold change and things like that, yeah, that would be showing you the difference, and then you can see whether the same difference is observed in TCGA. But you cannot compare directly the values, right? So if you have 10 in RT-PCR and you have 10 in RNA-seq, those would mean different things.
Okay. So will it be a qualitative comparison or a quantitative? Means if I see
Yeah,
any gene to
qualitative.
be I mean, if I see any genes high in the TCGA data, and if I see the same gene also overexpressed in the RT-PCR data, and if I want to compare the fold change, will it be a qualitative comparison or a quantitative? If it is quantitative, is there any mathematical formula so that I can compare these two different units? And the RT-PCR has got a
No,
different unit than the.
no, they're different in their, so they are different in what exactly they measure. So you would not want to compare the absolute values. You would want to compare qualitatively. Higher and higher and lower there would mean that they are comparable. But you cannot say that 10 here and 10 there are, somehow related.
Okay, thank you.
Yep. So just to finish with this idea of exploring your data, I hope that this is explaining to you also in a visual way that the fold change and the significance is actually going to be dependent on how you prepare the data. So aligning, making sure that your distribution is comparable within samples is going to help you eliminate false positives in terms of differentially expressed genes. So if we go back to differentially expressed genes, let's just take a look again at what are the numbers here, right? So you can see in the raw, you have a lot of them. In the logarithmic scale, you have a little bit less. And then quantile normalized, you have significantly less. So this would be the most reliable, but of course, you might want to kind of take the middle between these two, probably, when you want to identify those genes that you want to explore. And here, just to give you something to work with, you can take a look at these. So we're looking at normal-like and cloud and low genes, right? So what are, or cloud and low samples? What are cloud and low samples? Well, cloudin is a gene, CLDN, actually many different numbers. So CLDN 1 to 16. And you can go through them and see which ones you can find where. So I highlighted here a few examples. These genes, you can try and look at the differentially expressed, whether they are differentially expressed according to log value and according to quantile normalized data. So this is what I had for today. I hope you're getting a sense of how you can explore your data before jumping to conclusions about what is differentially expressed and what is not. And we will take a look at also how this impacts our downstream analysis. We'll take a look at, we can explore data in more detailed ways using PCA and different clustering techniques. Because of course, when we do differential gene expression, we can just compare two groups, and that is a big limitation. But if we want to do a more detailed comparison, we want to look into what other patterns and variations are in our data, we have to rely on some machine learning tools. And so we'll do a little introduction to machine learning and some of these methods next time. In the meantime, I hope that this lecture was informative to you for preparing your data and understanding your data and how the data really looks like by using some descriptive statistics and transformation methods like filtering, logarithmic scale, and quantile normalization. Before we disconnect, do you have any other questions? Okay, great. Well, again, for Kritika, we will take a look at your pipeline and what is going on there. I hope that everybody had a chance today to prepare and run their differential gene expression pipeline. And of course, we will see fairly soon what the result is. And then we will discuss further how we can manipulate and analyze our data and understand what's going on. Thank you everyone for joining today, and looking forward to speaking to you again soon.